Your brain on love lights up the exact same circuit as your brain on cocaine. Same neurons, same rush, every single time. So what if the thing we call love isn't romance at all? What if it's just brain chemistry, hijacking you on purpose? Let's set the rules first. We're not talking about liking someone. We're talking about the full chemical cascade: dopamine, oxytocin, serotonin, the three molecules that build, bond, and obsess. Could your brain really be getting high every time you fall for someone? Let's find out. Start with dopamine. It's the molecule of wanting, not liking. It doesn't reward you for getting the thing. It rewards you for chasing it. That distinction matters more than almost anything else in this story. Anthropologist Helen Fisher spent decades scanning the brains of people who had just fallen in love. She put them in fMRI machines and showed them photos of the person they were obsessed with, alongside neutral photos of acquaintances. One region exploded with activity every single time the photo of their loved one appeared: the ventral tegmental area. It's a tiny cluster of cells deep in the midbrain, barely bigger than a grape. It's also the exact same region that fires when someone uses cocaine. Think about that for a second. Falling in love and using a stimulant drug share the same ignition switch. Fisher's research found this pattern held across cultures, across continents, across ages, from newlyweds in the United States to couples in rural China who had never seen a brain scanner before. New love doesn't just feel like an obsession. Neurologically, it behaves like one. That's stage one. But dopamine alone doesn't explain why people stay together for decades. Wanting gets you through the door. It doesn't explain the marriage. Here's where it escalates. In 2000, neuroscientists Andreas Bartels and Semir Zeki at University College London ran their own scans on people who described themselves as deeply, intensely in love. They found something strange happening in the background. Regions of the brain tied to critical judgment, social assessment, and fear processing went quiet. Not just less active, measurably suppressed compared to the resting baseline. Your brain, when it's in love, partially shuts down its own bullshit detector. That's not a metaphor, and it's not an insult. That's what the scans actually showed. So the chase gets you in the door, and your judgment gets dimmed on the way in. Is that romance? Or is that a con? Your own brain is running on you without asking permission first. It gets stranger from here, because wanting isn't the same as bonding, and bonding needs a completely different chemical system, one that doesn't care about the chase at all. Enter oxytocin, sometimes called the cuddle hormone, which badly undersells what it actually does. Oxytocin is released during touch, during eye contact, during sex, during childbirth, even during a long hug with a close friend. It's the molecule that turns a stranger into someone whose absence physically, measurably hurts. Neuroscientist Larry Young built much of his career studying this in prairie voles, small rodents that happen to be one of the only mammals on Earth that form lifelong pair bonds. Young found the mechanism was almost entirely oxytocin-driven. Block oxytocin receptors in a prairie vole's brain, using a chemical antagonist, and it stops bonding with its mate completely, even after mating. Boost oxytocin artificially instead, and a vole will bond with almost any nearby partner, whether it's the right one or not. Humans obviously aren't voles. Our social lives are more complicated. Our choices are shaped by memory, language, and culture, in ways a rodent's never will be. But the underlying receptor system is strikingly, almost eerily similar. Every time you hold someone's hand, every hug that lasts more than 20 seconds, every quiet moment of sustained eye contact, your brain is quietly wiring your nervous system to that one specific person. It's not poetic license, it's chemical infrastructure, laid down one interaction at a time. So now you've got dopamine pulling you towards someone, and oxytocin cementing the connection once you're close enough to touch. That should be the whole story. It isn't. There's a third player, and it's the strangest one of all. Because here's the part almost nobody talks about at dinner parties. New love doesn't just feel obsessive as a figure of speech. It might actually be, biologically, a mild and temporary form of obsession. In 1999, Italian psychiatrist Donatella Marazziti ran a study comparing three groups of people: a group newly and intensely in love, a group formally diagnosed with obsessive-compulsive disorder, and a healthy control group with neither condition. She measured their serotonin transporter levels in blood platelets, a marker closely tied to mood regulation and compulsive, looping thought patterns. The results surprised even her. The newly in love group and the OCD group had nearly identical serotonin transporter levels. Both groups sat far lower than the healthy control group, with no meaningful statistical difference between the lovers and the patients. In plain terms, new romantic love and clinical obsessive-compulsive disorder showed the same biochemical fingerprint, at least for a while. That's part of why you can't stop thinking about someone in the early stages of falling for them. That's why their name lighting up a phone screen can scramble your entire afternoon of work. It isn't only infatuation talking. Serotonin dysregulation may genuinely be doing some of the driving, whether you're aware of it or not. So here's the real question this all builds to. If dopamine is chasing you, oxytocin is binding you, and serotonin is quietly obsessing you, is any part of falling in love actually a choice you're making? Let's bring the science together, because the full answer is more interesting than pure chemical puppetry. Researchers now generally describe romantic love as three overlapping, partly independent systems. Lust, driven mostly by testosterone and estrogen, evolved to get you looking in the first place. Attraction, driven by dopamine and norepinephrine, the can't eat, can't sleep, can only think about you phase. And attachment, driven by oxytocin and vasopressin, the slower-building, longer-lasting glue. These systems don't run forever at full intensity, and that turns out to be the whole point. Fisher's research suggests the dopamine-fueled attraction phase, the genuinely obsessive stretch most people call the honeymoon phase, typically fades somewhere between 12 and 18 months into a relationship. Not because love ends there, because your brain literally cannot sustain that level of neurochemical intensity indefinitely without cost. It would be exhausting to run at that pace, possibly even dangerous, in terms of sleep, appetite, and stress hormones. Your body downshifts the chemistry on purpose, like an engine designed to cool itself. What replaces it, when a relationship works, is the oxytocin-driven attachment system taking over as the dominant chemical signature. Calmer, steadier, less like a drug high, and more like a stable chemical baseline built jointly between two specific nervous systems that have spent months, or years, wiring themselves to each other. That's the twist hiding inside all this data. The chaos of early love is designed to burn itself out. What's supposed to survive that burnout is something far quieter and far more durable. So was the opening claim true? Is falling in love really the same as a drug rush? For those first frantic months, the evidence says yes, startlingly so. Same brain region lighting up, same suppressed judgment centers, same obsessive biochemical signature as a diagnosable disorder. But it's a rush your own biology, built on a built-in timer, engineered from the start, to hand control off to something more durable once its job is done. Here's the modern mirror worth sitting with. We live in a culture that's become obsessed with the chase itself. Dating apps are built almost entirely around dopamine, the swipe, the match notification, the uncertain, intermittent reward that keeps a thumb moving. That's the exact same circuitry Helen Fisher found lighting up in new lovers decades before smartphones existed. It's also, not coincidentally, the exact same circuitry casinos have spent a century learning to exploit. Maybe that's part of why modern dating can feel so exhausting for so many people. We've built entire industries around keeping people locked inside the dopamine-fueled wanting stage, without ever reliably routing them toward the slower oxytocin-driven bonding stage that's supposed to follow it. Endless chasing, rarely landing anywhere long enough for the deeper chemistry to kick in. There's one more piece worth knowing, because it changes how you might think about your own past relationships. None of these three systems are perfectly synced to the same person at the same time. You can feel intense lust for someone, deep attachment to someone else, and obsessive dopamine-driven attraction toward a third person, all in the same week. Fisher has argued this is exactly why humans can love one partner deeply while still feeling a jolt of attraction elsewhere. The brain didn't evolve one unified love system. It evolved three separate ones, running on different timelines, occasionally pulling in different directions at once. So think about that the next time your heart races over a name lighting up on a screen. Somewhere behind your eyes, right now, a tiny midbrain structure barely the size of a grape, is doing exactly what it did for every human being before you, and exactly what it will keep doing for every human being after you're gone. Your brain on love really does light up like your brain on a drug. The only real difference is that this is one hijacking your own biology voted for, every single time.